My name is Marcel Sanchez, and today we'll be taking a look at the molecular genetics of viruses. What this means is that I will be explaining how a virus works and what it's comprised of. So without further ado, let's get started. First off, what is a virus? Viruses are parasites of cells, essentially. A typical virus penetrates a cell, takes control of its metabolic machinery, and assembles hundreds of new viruses that are copies of the cell. Viruses are communicable agents that cannot replicate without the presence of the host cell. So penetrating the host cell, reproducing, and staying away from the defense system of the body are the main survival points of viruses. The host cell is usually destroyed in a process called the lytic cycle, but we'll address that later on. So what is metabolic machinery? You might be wondering what metabolic machinery is. Metabolic machinery is the components in an organism that take part in the metabolism or the chemical processes that occur in an organism to maintain the life of substances and macromolecules. So continuing on, viruses are very specific for the kinds of cells they will parasitize. What that means is that a virus will only attack a specific cell. For example, bacteriophages or phages are viruses that only attack bacteria. Phages will not attack any other type of cell. Now, let's get into the overall structure of a virus. A virus is comprised of a nucleic acid head and capsid, or a protein coat. Capsomeres assemble to form the capsid. An envelope may surround the capsid of some viruses. Envelopes contain phospholipids and proteins from the cell membrane of the host. The nucleic acids can contain RNA or DNA, but not both. You may be wondering what the difference between RNA and DNA is. DNA is the major storage for genetic codes that contains information for the functioning and advancement of all organisms. DNA is usually found in the nucleus of a cell. RNA or ribonucleic acid is a nucleic polymer acid that performs a significant role in translating the genetic code from the DNA to protein products. It is found in the nucleus and cytoplasm. It is usually a single-stranded molecule with shorter nucleotide chains. The sugar present is ribose. All in all, DNA is usually double-stranded, while RNA is usually single-stranded. So, replication viruses. Now that we've had some background knowledge on viruses, let's get into how these viruses replicate. There are two different ways that viruses replicate, either through the lytic cycle or the lysogenic cycle. Now, let's take a deeper look at both of these processes. So first off, the lytic cycle. I mentioned the lytic cycle earlier, but now we get to take a deeper look at this process. The lytic cycle is one of the two cycles of viral reproduction, the other being the lysogenic cycle. The lytic cycle results in the destruction of the infected cell and its membrane. A key difference between the lytic and lysogenic phage cycles is that in the lytic phage, the viral DNA exists as a separate molecule within the bacterial cell and replicates separately from the host bacterial DNA. The location of viral DNA in the lysogenic phage cycle is within the host DNA. Therefore, in both cases, the virus replicates using the host DNA machinery, but in the lytic cycle, the virus is a free-floating separate molecule to the host DNA. Viruses of the lytic cycle are called virulent viruses. In the first stage, called penetration, the virus injects its own nucleic acids into a host cell through the cell membrane. After that, the viral acids form a circle in the center of the cell. The cell then mistakenly copies the viral acids instead of its own nucleic acids. Then the viral DNA organize themselves as viruses inside the cells. When the number of viruses inside the cell becomes too much for the cell to hold, the membrane splits and the viruses are free to infect other cells. So, we just covered the lytic cycle, but now let's take a deeper look at another way viruses replicate. Certain viruses remain active within their host cells for a long period of time without replicating. This cycle is called the lysogenic cycle. The viruses are called proviruses because they do not bring immediate death to the host cell. The virus remains inactive until external stimulus, radiation or certain chemicals, causes the virus to begin the destructive lytic cycle. An example of the lysogenic cycle occurs in HIV infection or rabies, for example. So during the lysogenic cycle, the original virus attaches itself to the target cell and penetrates the cell membrane, just like in the lytic cycle. But unlike the lytic cycle, during the lysogenic cycle, the virus mixes its genetic instructions into the host cell's genetic instructions. When the host cell reproduces, the viral genetic instructions get copied into the host cell offspring. This can go on and on until some outside stimulus causes the virus to move into the lytic cycle and burst out of the host cell. That is why during the first 6 months of the rabies infection, there are no symptoms prompting that it is rabies. That is also why during these first 6 months, rabies can be mistaken for the common cold or other minor sicknesses. This concludes this lesson on viruses, and I hope you learned a few new things about viruses. Thank you for watching, and as always, have a nice day.